Hello everybody, and welcome to my new channel, Science Abounds, Science at Home, where we're going to discover and do some lab magic right in the comfort of your home. Yes, you heard me correct. We're going to be doing chemistry experiments, biology experiments, some physics experiments, right from the comfort of your own home. In these videos, I will be performing these home experiments, demonstrating scientific principles and scientific reactions. And I will be doing it here in my house, in my home office. As you can see, I'm sitting on my bed. I have a lab table, a study table to do my experiments on. You really don't need a lot of space. You can pretty much do home experiments in your kitchen, in your garage, in a bathroom, your living room. And during these videos, different videos will be shot in different parts of my house just to demonstrate the fact that you can do home experiments literally anywhere in your own home. The experiments I'm going to be using are from a variety of sources, but I'm going to be starting out with using kits from Mount Chemistry or Mount Science. Okay, and I will put a link in the description so you can yourself go to their website and order these experiments so you can perform them along with me in your own home or anywhere that you want to do your experiments. They are a subscription service. They are not sponsoring my videos. I'm just recommending these. These are phenomenal kits that demonstrate chemistry principles, and they deliver kits twice a month. You get 2 experiments a month. And when you first take out a subscription with Mount Science, you will get a starter kit, which is what I have here. And basically, the starter kit contains everything which is generally found in every experiment that you do, including glassware like a beaker, goggles, safety apparatus, conical flask, funnel, things that you're going to use in nearly all your experiments, plastic caps, candles, and they use candles to provide source of heat. They provide a burner, manual apparatus where you put the candle in like so, and you have a table or a surface to put glassware on top if you want to warm up solutions required for the experiment. It's in place of a gas Bunsen burner, which you simply cannot have in your home. So you have all the starter elements in the starter kit. You have tubing as well, everything that you need to get started, but also the kits that you get each month, like for example in here, this is about sulfur and nitrogen dioxides, which we will be doing in a future video. And each kit contains all the needed chemicals and additional consumables and apparatus that you require to do that specific experiment, along with the consumables that can be found in your starter kit. These kits are aimed from ages 10 and above. Some state 12 and above. I always recommend anyone below the age of 12, 11, you should have an adult present to do the experiments with you and to make sure you're doing them safely. All these experiments are safe to do. The reagents are in very small containers. There is enough usually to do the experiment twice, if not more. And they're in small amounts, so it's very safe. There's no huge containers of flammables or poisons or anything like that. we want to do home experiments safely but effectively. And all the experiments that I'm going to do require no special laboratory environment, so that you can enjoy science without having to worry about all the dangers of that science can involve. So, what are we going to be doing? Well, like I said, I am a scientist myself. I have scientific degrees. I am a practicing clinical scientist working in a hospital laboratory. And I'm going to be demonstrating scientific principles and scientific reactions through experimentation. Each week I will be demonstrating a new principle, a new reaction, highlighting science, but it will be predominantly chemistry at the beginning because that's my passion. But over the course of time, I will be doing biology, we'll be exploring the microscope, looking at things, beautiful things under the microscope that you can't see normally, the details, the intricacy of biological elements with the naked eye. So we're going to explore microscopy and the world of the microscope and biology. We will do some physics experiments, some experiments relating to medicine, forensics. It's limitless to what you can do at home. So, what are we going to do today? We are going to start with our very first home experiment. Today, I'm going to introduce you to pH. pH. What is pH? I'm sure everyone's heard of it. I'm sure everyone's heard of acid, alkalines, basics. Well, we're going to explore those. And we're going to explore how pH works, how we can look at acidic compounds, and how we look at alkaline compounds. And in fact, this video and the next video are going to be talking about pH because it is fundamentally very important to science and everything about life. So, in this experiment, we're going to be using, we're not going to be using the burner, so let me put that aside. It's nice you get a tray that comes with a starter pack, and this is very useful to have to do your experiments on, to contain any spillages. So if you do spill any chemicals, it's easily to wipe off and wash off. So there's only 3 compounds or 3 reagents that I'm going to be using in today's experiment. I'm going to be using sodium carbonate, sodium hydrogen sulfate, and I'm going to use thymol blue. Thymol blue is the indicator, is our pH indicator. And the basis behind pH indicators is that they change color depending on the pH of a compound or solution. And there's usually a pH, what we call a pH scale. And this scale usually goes from 0 to 14 or 1 to 14. And 1 or 0 being the most acidic of compounds or solutions, and 14, 15 is the most alkaline solution or compound. And we're going to look at that scale, but in the next video we're going to look how the scale ranges throughout different types of acidity, different levels of alkalinity, and so on. So more about that in the next video, but in this video I just want to demonstrate how color changes of an indicator based on the acidity and the alkalinity of a compound. And I'm going to be doing these reactions in a petri dish. Yes, you heard right. It's a petri dish is obviously something that we are familiar with in microbiology experiments where we grow bacteria and so on. But petri dish have multiple functions. We can do tissue cultures. We can put things on the petri dish to look at in microscopes, as well as chemistry experiments as well. So the experiment is going to take place in the petri dish. So let's get started. Okay, so what I'm going to do first is I'm going to add just a few drops, 3 drops of sodium carbonate. And as far as safety is concerned, I would strongly advise the little students to wear safety goggles and gloves. So I'm going to open my sodium carbonate bottle, and they are safety. So with my sodium carbonate, I'm going to add 3 drops of solution. It comes in a liquid form, and I'm going to add 3 drops to the petri dish. Oh yes, you need to burst. Let me start again. To pierce the container. So I'm going to add 3 drops. 1, 2, 3. 3 drops. It doesn't matter if you do more. And next what I'm going to do is I'm just going to make a solution and dissolve and add some water. Okay, and I'm going to mix that up. Okay, let me add a little bit more water because I want to cover the whole of the petri dish. All right, so let's add it on. Give that a little mix. So now what I'm going to do is I'm going to add my indicator. I'm going to add the thymol blue. The thymol blue is a dark blue solution. Okay, I'm going to prick this as well, like so. And watch what happens when I add this blue solution to the sodium carbonate. Okay, it turns blue, which is the color of thymol blue. So we can mix this up to equilibrate it, so we have a blue solution. So the blue didn't really change. The thymol blue color didn't really change. It remained the same. And that's because sodium carbonate is an alkaline solution. It's an alkaline compound. It's basic. It's an alkaline compound. What does that mean? Basically, alkaline compounds or basic compounds in solution produce hydroxide ions, OH ions. And it's the OH ions that make a compound alkaline. And thymol blue changes structure under different pH conditions. And with this structure change comes with a change in color. So right now we've got sodium carbonate in our solution and it's blue. So right now we have an alkaline solution. But what happens when we add sodium hydrogen sulfate, the other reagent that is part of this experiment? Well, again, I will remove the lid. I will pierce the top. And let's see what happens. Are we going to see a color change? I hope so. Oh, look at that. Look at that. So I can keep adding. You see when I add it, it turns red, orange, red. Look at that. So the thymal blue is changing color because acidic compounds in solution produce hydrogen ions. And the more solution of the acidic compound I'm adding, it's turning red, orange more and more as the solution that we see is going from a basic solution, an alkaline solution, which was blue, to an acid solution, which is a red-orange color. Because the hydrogen ions are dominating now, because I've added more and more of the acidic solution. So now there's no more basic solution, or there's predominantly acidic in solution now. And this is where we got a color change. So what happens if we add more alkaline solution? Let's have a look. Let's see what happens. Let's experiment. Oops, I can't open the bottles. All right. So let's see what happens. And there you go. The area that we've applied the alkaline solution, you see it changing blue. But right now, it's still a very strong acid solution, so it's not dominating. But I could add more and more, like so. I want to produce more and more hydroxide ions to make the solution more and more alkaline. So it's neutralizing the acidity, the acid solution. And right now it's in equilibrium. So I have equal parts of alkaline and equal parts of acid. So I get a mixture of blue and a mixture of red-orange in color. Okay, so if I keep adding sodium carbonate and keep adding more and more hydroxide ions, then I'm going to overpower the hydrogen ions and make the solution more alkaline. Okay, that was a very simple experiment to demonstrate a very powerful scientific principle, scientific reaction, pH indicator. This is very important in the world, that we can identify acidic compounds and alkaline compounds and understand how by a simple pH indicator we can test very quickly to determine whether a compound is acidic or alkaline. This is used in everyday purposes. We measure, for those who have swimming pools and who have the luxury of having swimming pools in your backyard, your pool guy or your parents have to monitor the pH of your pool to prevent algae from growing and to keep it hygienically clean. You have to measure and monitor the pH. So they do that. They monitor the water every couple of weeks, and they use a basic simple pH indicator. They may even use Thymal Blue, but there are other solutions that are available that are even more sensitive to pH fluctuations, and we will look at those later. pH is very important in the environment. In rainwater, we monitor rainwater pH for acidity because acidity can cause erosion here on the ground. You may have heard of acid rain, and this is due to the presence of compounds such as sulfuric acid and nitric oxides, which are from the which form in the atmosphere from waste that is emitted by numerous industry, like big turbines, electricity burners, and things like that. They all produce waste gases, waste compounds, and they go up into the atmosphere and they form acidic compounds which then come back to the ground in the form of acid rainwater. And that leads, like I said, can lead to erosion over time. Also, pH is very important in biological functions. In your own body, different parts of your body are at different pHs. In your mouth, it's an alkaline environment or a basic environment. You don't want acid in your mouth. You have enzymes that can be found in the saliva which help neutralize and keep the mouth neutralized, and it prevents corrosion from acidic compounds. That's why we tell you not to eat or drink acidic foods, acidic drinks, because they can corrode the enamel in your mouth. And sometimes the enzymes in your saliva cannot fight quick enough the acidic compounds that are in your mouth, and that's why you get corrosion of the enamel. In your stomach, very, very acidic environment. In fact, you have hydrochloric acid at very concentrated levels in your stomach, all to break down food and to help with the digestive process. And you actually, for those who have indigestion or heartburn, you feel the pain as a result of the acid attacking the lining of your stomach. And there are some medical conditions, for example, a peptic ulcer, which is the lining of your stomach has worn away and the acid that your stomach produces is attacking your own stomach. And that can be very painful. If anyone's experienced an ulcer, it is very painful indeed. And then as the food travels down into your intestines, it becomes a basic or alkaline environment where the acid is neutralized and the enzymes found in your duodenum, in your intestines, produce an alkaline environment. Right, I hope you enjoyed that little experiment. It was just very basic, very simple intro to my video. My video is for any age, from somebody in elementary all the way to adults, hobbyists who are just interested in science, learning about basic principles of science, reactions, and what they can do and how they can experiment in their own home. So my videos are aimed to be fun, but they are educational, but they are not going to be academic in nature. They're just going to be, I'm keeping it to be fun, basic science. I'm teaching the fundamentals. The videos are going to be raw. There's no rehearsing of my videos. I'm just going to come in, do my experiments, show you how easy it can be done, and the exciting results that you can see and why it can be fun. But at the same time, I want you to learn a little bit about the background of what is happening during the experiments. Please reach out to me. At the end, you'll have my email. Please leave comments, good or bad. What can I do better? What needs improving? This is my first video, so it's going to be raw and rough. But please do comment. I want to hear from you what you want to see on my videos, what kind of experiments you want to do. I'm not here to make explosions. I'm not here to show you how to make bombs or drugs or things like that. That is not the purpose of my videos. So please do not ask for those kind of experiments. So I hope you will come back. My videos are going to come out once a month at the moment. And I hope you will join me next time where we will discover lab sites in the home. Thank you everybody for joining me. Bye-bye.